Hi all, Dr. Clark here again, and today for general biology we're going to talk about chromosomes. So remember, last time I gave you kind of an overview of what mitosis is, or the cell cycle, in both prokaryotes and eukaryotes. Now we're going to talk about chromosomes, or the way at which organisms package DNA, especially eukaryotes. So we're going to talk mainly about eukaryotic chromosomes today. Now, things are changing. We're learning a lot more about chromosomes than we ever did. And so, in your textbook, and the way I normally teach chromosome morphology just means the way they look. The way I normally teach it is I talk about nucleosomes and histones and the wrapping of DNA around the histones to form this kind of coil up chromosome. And I'll still teach that. That's called the textbook model. And I'll still teach that because currently the evidence out there is not so decisive that we need to move away from the textbook model. But there's also a new model, which is called the condensed mediated model, or condensed mediated, model. And that model is basically indicating that researchers have been able to indicate that if you remove the histones, which we'll get to this, and it'll be much clearer on your part when we get to this. But if you remove the histones, which are proteins that the DNA, the chromatin, wraps around, right? If you remove them, the DNA still wraps in the same form. Okay? I mean, if you look at these two models, this is one with histones, this is one without, and the formation, the compactedness is pretty much identical. I mean, it's a little bit looser, it's a little bit different shape, but for the most part, regardless of the histones, regardless of the proteins, DNA is folding and condensing into these, tightly packed chromatin molecules, which we call chromosomes, which is really cool, because we always thought histones were the secret, but there's got to be something else that's going on, because without histones, they still condense. We'll come back to this. I just wanted to kind of give you this piece. This is new information that's coming out, and I like to, try to update you as much as possible, even though the textbook won't probably update this kind of information for a long time, if ever. I'll try to provide you that insight later. All right, so chromosomes. Now, we often think about chromosomes from the standpoint of humans, and you know that we have 23 different chromosomes, and that ultimately means we have 2 copies of each one of those. One copy that you inherited from your dad, one copy that you inherited from your mother, and that gives you 46 chromosomes. Okay, so you can number your chromosomes 1 to 23, and you have 2 copies of chromosome number 1, 2, 3, all the way to 23. We are a diploid individual. Okay? We have 2 copies of every chromosome. Now, there are lots of other organisms on this planet that are diploid, but not all of them. There are probably far more organisms on this planet that are not diploid than there are that are diploid. And on top of that, when we talk about eukaryotes, we talk about an estimate of between 10 and 50 chromosomes. Remember, prokaryotes are just typically one chromosome. But most eukaryotes are between 10 and 50. But that doesn't mean that you can't find exceptions to that. You can't find, crabs that are out at 200. You can't find bulldog ants that are down at 1. There's lots of variation of organisms. And that just depends on also what we're talking about. Are we talking about the number of different chromosomes between 10 and 50? Or are we talking about the total number of chromosomes? And if that's the case, this number is wrong. 10 to 50 is very wrong. Okay? And I'll show you why in a little bit. All right, but regardless of that, chromosomes are highly useful in an organism because chromosomes are the condensed form of your DNA. And we think that it probably condenses into the typical shape that we consider an X, or if we're talking about birds, the Z. And we'll get to this also. Not all organisms have the X-shaped chromosomes. But that condensed version, we think that it probably evolved because when the nuclear envelope, the membrane that goes around the nucleus, when it dissolves, that DNA is no longer protected, okay? And it's just hanging out there doing nothing. And if it's in long, thin filaments with no condensing, it can break, it can be damaged. So chromosomes probably evolved this wrapping of chromatin or DNA material around histones, around proteins to make them condensed. Probably evolved because you know, the envelope's gone and that DNA still needs protection. Now, chromosomes occur in what we call homologous chromosomes or homologous pairs. Normally, they occur in this, and I'll talk situations where that's not true in a little bit. Now, homologous chromosomes, especially in diploid organisms like ourselves, you have one chromosome from your dad, one chromosome from your mother. Okay? These are identical chromosomes, so it's chromosome number 1 and chromosome number 1. But the genes on those chromosomes can be varied. So it's the same gene. So let's say we're talking about chromosome number 1 and chromosome number 1 for your mother. And let's say we're talking about a protein that allows for you to digest milk. So a lactase protein. Maybe your dad gives you the recessive trait, which means that he gives you a version that's different than your mother's, which maybe she gives you the dominant trait. And all that means is that that gene is not the same. It still codes for the protein lactase, both of them, but just a little bit of variety in that coding. So the protein is going to look a little bit different, have a little bit different shape. Okay? And that's all we're talking about when we talk about genetic diversity, is we're not talking about having different genes often. We're talking about having different variations of the gene. Okay? And we'll talk a lot more about that when we talk about Mendelian genetics. So again, if you have 2 copies of every chromosome, then you're considered diploid. If you have 3 copies, you're considered triploid. If you have 4 copies, you're considered a tetraploid. And 5, a pentaploid. And on and on and on. And there's a huge variety of organisms that have way more than 2 copies of every chromosome. Now, we already know this because we talked about it before. So during the cell cycle, after the G1 phase, the cell will enter the S phase. And the S phase is the copying of the DNA. So in our S phase, let's talk about human cells. When your cell moves from the G1 to the S phase, it's taking, each cell has 46 chromosomes. It's taking those 46 chromosomes and it is copying all 46 chromosomes. So at the end of the S phase, you actually have what we call 92 chromatids. Okay, so you actually have 92 individual chromatids, and we'll talk about what's going on after that. So you go from 46 chromosomes to 92 chromatids. You haven't really changed any of the genetic information, you just doubled it all. Now those chromatids are going to be joined together with a centromere. And centromeres normally occur pretty close to the center of the chromosome, and it depends on the type of chromosome, it depends on which chromosome we're talking about. But it normally holds the 2 chromatids together, And there's also another force that happens to hold those together, which we already talked about, and that's cohesion, and the attraction of molecules to each other. That's going to hold it a little bit too, plus the centromere. So again, like I said before, if we have 23 pairs or 46 chromosomes, and you go from G1 to the S phase, and at the end of the S phase, you've copied all 46, so now you have 92 chromatids. This is essential that this happens, because we're going to take those 92 chromosomes, or chromatids, and we're going to divide them back out into two separate cells. So you're going to have one cell with 92, and you're going to divide it into two separate cells, each with 46. You're going to return the original number. And that's the importance of mitosis and meiosis in particular, is that you're reducing, or you're starting with more chromatids and you're reducing it to the original number. In the case of meiosis, we'll see that we reduce it even further than the original number, so we can return the original number through sexual reproduction. We'll get to that in a later section. All right, so here's the homologues. So let's say that purple is for the parental, or the father, okay, or the paternal, and green is for the maternal. Okay? And so this is what you have. These are chromosomes. And then after the S phase, you're going to have a copy of each one. Okay? So this you might have inherited from your father, and this you inherited from your mother, and now you've copied each one. Okay, I hope that makes sense. So this could be chromosome number 1, 2, 3, on and on and on. Probably not chromosome number 23, because that's a sex chromosome, so it's going to look a little bit different. But nonetheless, almost all the chromosomes, pretty much the exact same situation to make homologous chromosomes. All right, so let's look at a karyotype. So this is a karyotype. This is a human karyotype. You can see all the different chromosomes, and there you can see chromosome number 23. This is an X and a Y, so that makes this individual that's karyotyped. This makes this individual male, okay, that Y chromosome. Now, we can look through these chromosomes and we can see that they're very different. Now, I don't know if you can tell, but some of them you can see where the centromere is, like here and here, and this centromere is here and here, okay, and it's up here. Okay, we'll talk about centromere placement. As for like some chromosomes, like chromosome number 21, they're going to be acentric centromere placement, so it's going to be towards one tip, or closer to one end, so it's going to cause a elongated end, okay, which we call the Q zone, and a short end, which we call the P zone. And we'll talk about this, don't worry. But this is the chromosomes of a human. One is from the dad, and one is from the mother. Okay. Now we can compare chromosomes, and we've done this many times. We can compare them based on their size, their shape, where the centromere is located at, and now, with modern technology, we can compare how many different genes are on each chromosome. So now we have the capability of selecting genes, and here you can see regions that turn different colors, or regions that are being expressed and regions that are not being expressed, and whatnot. So we have those capabilities now, far more than we did in the future, but even in lab, you guys saw how we can karyotype. We can splat chromosomes open, and then we can match them based on their size, their shape, and where their centromere is at. And that's how we originally figured out that humans have 23 chromosomes and that they have 2 copies of each. Okay, so 23 pairs. All right, so let's talk about a little bit about structure. I just kind of mentioned it. So there is four main structures or four main types of chromosome or types of centromere placement on chromosomes. So you have centromeres. The telecentric chromosome would be a centromere that is at the very end of the chromosome. So there is no P arm to the chromosome. Okay? And these do not, at least as of we know right now, these do not exist in humans. Okay? Lots of other organisms have telecentric chromosomes, but we haven't found them in humans. So there's always at least a little P arm, P zone on our chromosomes, human chromosomes. Telecentric, no P zone. Okay. Acrocentric means that there is a P zone, but it's very short. Humans have these. Okay. Chromosome number 21 is an example. Okay. And they have a long section, which we call the Q zone. And you know, the centromere is just offset. It's offset from the center, and it causes that one long section, one short section. I'll show you a diagram of it in just a second. Submetrocentric just means that it's off center a little bit, so slightly asymmetric. And so there's a P zone that might be a little shorter than the Q zone, or vice versa. And the metrocentric means that the centromere is nearly perfectly in the middle, and the P arm and the Q arm are as close to equal as possible. So what does this look like? So here's an example. That's the centromere. Remember, centromere is what's holding the sister chromatids together. And so that centromere is there. Telecentric means that there is no P arm. Acrocentric, a little bit. Submetrocentric, a little bit more. Metrocentric, the same amount as Q. So P arm stands for petite. Okay? And so the first individual who discovered differences in centromere placement called the arm the P arm, or the petite arm, the smaller of the two arms. He originally called the other arm the G arm, which was, a greater arm or greater than arm. But then it got changed to the Q arm. And so the P arm represents the short region, and the Q arm represents the long region. So sometimes you'll hear or read papers or hear people talk, they'll say, well, P1 or P2 or P3 or something like that was moved or removed, or that's where the gene's located. That's what they're talking about. They're talking about the region, the short portion of the chromosome, and they're talking about a specific region, which we call loci, a specific region on that chromosome. That's what they're talking about. Okay, so chromosomes themselves are composed of what we call chromatin, which is DNA and protein. And so you have some proteins that are incorporated with the DNA, kind of I don't want to say in cases, but it binds with the DNA. So remember that DNA has a slight negative charge, and that's due to the phosphorus that's on the backbone. And so they have a slight negative charge, and most proteins are going to have a positive charge, okay, or a neutral charge. But a lot of proteins will have a positive charge. So the protein's attracted to the DNA. So there's always going to be some association with proteins and DNA. And that makes up chromatin. Now there's also other segments, other nucleotides that are involved and incorporated. And so you'll have some RNA, whether this be messenger RNA, or in some cases, depending on where the DNA is at, you might have some ribosomal RNA that's associated with the chromosome. But you're going to have some other RNA that's associated with it and associated with those proteins. Now remember that when we are talking about human chromosomes, when we're talking about human DNA, our genome, if you take all 23 chromosomes and you stack them together and you line them up and you stretch them out, okay, it's 3 billion nucleotides long. So you can imagine that you have to cut that 23 times, and different chromosomes are different sizes, obviously, but it's a lot of nucleotides. It's a long string of chemical bonds that make up our DNA. Now, DNA, like I said before, this may change in the future, the importance of this. Well, we do know it's true that DNA coils around proteins that are called histones, and those histones have positive charge, and that's associated with the negative charge in the phosphate group of DNA, like I said before. And DNA, when it coils around these histones, it coils around roughly 8 histone proteins, and that makes up what we call a nucleosome. And then nucleosomes will wrap around other nucleosomes, and you get a very compact chromosome from this wrapping of chromatin and proteins and more proteins and wrapping them all together. And that's really what it takes to condense DNA to make it visible. Okay, we can't see chromosomes unless it's wrapped around a bunch of proteins. It's just not possible. You can't visualize it. So you have to have that DNA in this form. It has to be in chromosome 4 for us to actually visualize it and see it with the naked eye. Okay, we can see it without the naked eye, but to see it with the naked eye, and you'll see this in a lab that we do, it has to be wrapped around proteins. And even to see it under a microscope has to be wrapped around proteins, pretty much. You can see it with an electron microscope, and you can disassociate it with the proteins, but it's still a little more difficult to get a handle on that. And I'll show you some electron micrographs of that material a little bit. Okay? So here's what I'm talking about when we're talking about that organization, chromosomal organization. You have DNA that makes a chromatin section, so it wraps itself around histones, these positive charged proteins, and they wrap themselves around other material that forms a nucleosome. And then the nucleosome will form with scaffolding proteins and whatnot, and they'll form what we call a chromatin loop. So you get these kind of rosettes that form, and then those rosettes will all squish together to form that typical chromosome look. So it's a lot of protein that's mixed in to form these chromosomes. It's not just all DNA, it's not all genetic material. There's a lot of protein, and there's a lot of associated proteins even on the outside of chromosomes that help with either maintaining shape or allowing for these to stick together to help the centromere hold the two sister chromatids together. So unlike what we often think about when we think about chromosomes being X-shaped, the arms of the chromosomes are not actually crossing. So this is not one chromosome that crosses over here. Okay? This is one chromosome that goes this way. Okay? And then there's another one on the other side, or chromatid that makes up this. One chromatid on this side, one chromatid on that side that are bound by centromeres. So they don't actually cross like an X. They're just stuck together in the middle, so they make kind of a different shape. And most of them are not X-shaped when you actually get down to it. There are all kinds of shapes. Which brings me to this piece, the X, Y, Z, W, or other category. So when we look at mammalian samples and mammalian examples of chromosomes, we talk about ourself and our system, and that's an XX, XY system. So XX female, XY system is male, and that's pretty much the way it is for almost all mammals. At least all mammals that I'm aware of are on this system. If that's not true, I will definitely update this to make it true. All birds, however, are on a different system, which we call ZW or ZZ system. ZW is the female, ZZ is the male. So where in mammals the heterogenic chromosome pair is the male, in birds the heterogenic chromosome pair is a female. Now, when we look at the other vertebrates, the things that gave rise, and this provides a lot of evolutionary evidence, is that you would expect reptiles, which gave rise to mammals and birds, you'd expect them to have both systems. And indeed, they have both the systems and others. Okay, so aves, birds, have pretty much the ZWZZ system. Mammals, XX, XY system. Okay, reptiles have both ZW, XX, but they also have some of these other systems going on. Amphibians have other systems going on. Fish have a huge variety of systems going on. So when we just talk about sex chromosomes, we always think about it from a mammal standpoint, but I'm letting you know right now, there's roughly 4,000 mammals on the planet. That's it. 4,000 species of mammals. When we start talking about birds, there's 10,000. Reptiles, there's another 10,000. Reptiles and amphibians together, about 10,000. Fish, there's at least another 35 to 40,000 species. When we start talking about these other groups, we need to know that not everything is an XY system. Not everything is based on the human system that we know, that there's a lot of variety out there. And the cool thing about it is, especially in fish, check this out. Okay? So these are different species of fish. I'm not expecting you to remember them or anything. This is a cichlid, this is a hackfish, this is a wharfish. But when we start looking at them, okay, you can look at 2N means that they're diploid. What's their diploid number? So you can see that, you know, this guy's got 48 chromosomes, 50, blah, blah, blah. So they're all pretty even on the number of chromosomes that they have. You know, some dip. And you get some weird ones like this, or what's strange to us knowing the XY system. Look at this. If you're a male on this fish, you're sitting at 39 chromosomes. If you're a female, you're 40. If you're a male on this fish, you have more than you do if you're female, 53 and 52. So there's all kinds of things. But then look at what the sex system is. XXXY, okay, simple, we know that. ZZZW, okay, simple system. It's basically XY system, just reversed. This is a bird system. And then you get this guy. XXXXX, XXY. I mean, just bizarre systems. XX, XYY. Okay, these are systems that, show a great degree of diversity, and we don't know all the sex systems. We're discovering new sex systems. I mean, this one was discovered in 2009. We've known about that fish for a long, long time, but new fish that pop up, we probably don't know what their sex system is yet. So I'm just going to leave you with that. We'll come back and we're going to talk about now mitosis. So now I talked about the cell cycle, then I talked about chromosomes, and now I'm going to talk about mitosis. So how is it that these chromosomes get separated? What is the specifics on chromosome separation? Okay? Next time, that's what we'll talk about.